In our digestive system, we have small intestine and large intestine. The main function of small intestine is to transport food from stomach to large intestine, to absorb final products of digestion into blood and lymph vessels, and also to secrete certain gastrointestinal hormones. To maximize the absorption of small intestine, it has certain specializations, and we can see the specializations from serosa, submucosa, muscularis externa, and mucosa. For submucosa, in the initial portion of the duodenum, the submucosa contains cluster of cold branches, tubular glands, called the duodenal glands or Brunner's glands. Brunner's glands secrete alkaline mucus, and the acidic gastric secretion could cause erosion of the duodenal mucosa. The mucus helps to bring the intestinal contents to the optimal pH for pancreatic enzyme action. The lamina propria of the submucosa of the small intestine contain aggregates of lymphoid nodule known as Peyer's patches. For the muscularis externa, it consists of an internal circular layer and an external longitudinal layer of smooth muscles, whereas for the serosa, it is composed of a thin connective tissue covered by the mesothelium.
Small intestine consists of duodenum, jejunum, and ileum. For the mucosa, the precae, which is a series of folds, increase the surface area of mucosa by about 3 times, but a further 10-fold increase is obtained by the presence of finger-like projections, the intestinal villi. Between the bases of villi are small openings of simple tubular glands called the gastrointestinal gland or crypts of Lieberkühn. The epithelium of the gastrointestinal glands contains stem cells, some absorptive cells, goblet cells, Penneth cells, and enteroendocrine cells. For the absorptive cells, tall columnar cells basally located over nuclei. At the apex of each cell is a homogeneous layer called the striated brush border. The striated border is a layer of densely packed microvilli. Microvilli have an important physiological function of increasing the area of contact between the intestinal surface and the nutrients. For the absorptive cells, we can find the presence of precae, villi, and microvilli. Therefore, the absorptive cells are specialized for absorption of nutrient molecules produced by the digestive process. For the goblet cells, they are interspaced between the absorptive cells. They form mucus which protects and lubricates the lining of intestine. For the Penneth cells, they are endocrine cells with secreting granules which contain lysozyme in their apical cytoplasm. For the endocrine cells, they all contain secretory granules located basally to the nucleus. Upon stimulation, these cells release their granules that contain the hormone by exocytosis. For the stem cells, most of these cells migrate upwards and differentiate into absorptive cells. Others become goblet cells or enteroendocrine cells. For the M cells, they are specialized epithelial cells in the intestinal epithelium overlying the lymphoid follicles of Peyer's patches. M cells can endocytose antigens from the lumen of the small intestine and transport them into the underlying connective tissue, where they are taken up by the macrophage and lymphocytes. M cells differ from absorptive cells in that they do not have microvilli on their apical surface, but they have broader microfolds that give the cells its name. For the lamina propria, it consists of loose connective tissue with blood and lymph vessels, nerve fiber, and smooth muscle cells. The lamina propria fills the intestinal villi. For the muscularis mucosa, it consists of inner circular and outer thinner layer of smooth muscles.
Besides small intestine, the digestive system is also composed of large intestines. Firstly, the function of large intestines is the absorption of water and inorganic salts. Secondly, is the formation of the feces. And the third one is the production of mucus. The absorption of water is passive, following the active transport of sodium out of the basal surface of the epithelial cells. The mucus lubricates the intestinal surface and covers bacteria and particulate matter. Large intestines can be divided into three parts. Firstly is the cecum, second is the colon, and the last part is the rectum.
For the specialization, large intestine consists of four main layers, which is the mucosa, submucosa, muscularis externa, and serosa. The large intestine consists of a mucosa with no plicae except in its distal rectal portion. The thick mucosa contains intestinal glands which are longer and straighter than in the small intestine. These glands are characterized by a great abundance of goblet and absorptive cells and a small number of endocrine cells. The mucosa consists of epithelial lining, lamina propria, and muscularis mucosae. In the epithelial lining, there are full absorptive columnar cells with a striated border. The goblet cells occurs far more abundantly than in small intestine. In lamina propria, the reticular connective tissue is scarce, but there are many lymphoid cells and nodules. This is related to the abundant bacterial population in the large intestine. Whereas in muscularis mucosae, it consists of an inner circular and outer longitudinal layer of smooth muscles. The layer of submucosa consists of loose connective tissue. For muscularis externa, the inner layer consists of circular strands, but the fibers of the outer longitudinal layer are grouped together in three thick longitudinal bands called tenacoli. The mesenteric plexus is located immediately outside the inner circular muscle layer. For serosa layer, which contain mesothelium, there are small pouches of peritoneum filled with fat called appendices epiploicae.